As we've seen during the previous modules, aging is a process during which the function of many physiologic systems declines over time. In fact, as we have seen, many definitions of aging hinge on that very fact. The proper functioning of an organism, its organ systems, its metabolic activity, etc., is best described by the term homeostasis. Homeostasis is the concept that a biological system functions best within a certain set of parameters, such as a specific pH range, core body temperature, etc., 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 and that these sets of parameters are actively maintained by the body's organ systems. Think about it in terms of a waterway contained within a system of dikes. If the water level is too low, no ships can travel on that waterway, or irrigation of crops that depend on the water may be compromised. But if the water level is too high, it may crest over the dike and flood the surrounding area, resulting in damage. Thus, homeostasis of that waterway would be to keep the water levels neither too high or too low to ensure functionality of the waterway and its dependent areas. In other words, it's the Goldilocks principle. Thus, aging can also be viewed as a loss of homeostasis in many organ systems, or rather, the loss of the ability of the organism to keep maintaining said homeostasis. Maintenance of homeostasis is a very costly process. In terms of energy expenditures, it happens continuously, as disruptions of homeostasis occur almost constantly. Even the simple act of eating represents a huge disruption in homeostasis, as the body has now to deal with a sudden influx of nutrients that can change blood pH levels, insulin levels, neurotransmitter levels, and so on. In essence, a snowballing series of events that lead to changes in almost every system in the body. The body responds, for example, by making insulin, which removes dietary sugars from the bloodstream to bring blood sugar levels back to normal. Disruption of blood sugar homeostasis, by the way, is called diabetes. And as type 2 diabetes, we know it's clearly associated with aging and, in fact, is often considered a disease of aging. Events and conditions that disrupt homeostasis are what we summarize under the general term stress. And that's the topic of this module. As an organism ages, it loses its ability to properly maintain homeostasis. As the disposable soma theory states, proper functioning of somatic cells is not important in evolutionary terms, but only maintenance of the species is. Thus, the germ cells become important. With limited resources at its disposal and more stressors disrupting homeostasis over time, the inevitable result is that not all organ systems will be properly maintained. And in addition, the ability to respond to novel stressors diminishes at the same time. In the mid to late 80s, aging research began shifting its focus from a relatively theoretical exercise to the actual investigation of the molecular events underlying the aging process, in part spurred by the molecular biology revolution that was sweeping the natural sciences at the same time. With the advent of modern DNA technologies, it now became possible to isolate and investigate individual genes and the gene products. Using these technologies, researchers were now able to study the effects of individual genes on cell function, organ function, and organismal function, and in our context, longevity. The very first gene that was shown to extend lifespan of a model organism was isolated and characterized in the nematode C. elegans. It was aptly named H1. Since then, hundreds of genes with roles in longevity and aging have been discovered, characterized, and even been connected into functional networks, so-called pathways. What is shown here is the network of nematode genes that H1 is involved in. H1 is part of a signaling network that is known as the insulin/insulin-like growth factor signaling pathway, also abbreviated IIS. This signaling network gets activated when insulin or a very similar molecule binds to its cellular receptor. In C. elegans, that receptor is called DAF2. Through a series of interactions with other proteins, one of which being H1, a protein called DAF16 gets activated, which in turn acts as a transcription factor. A transcription factor is a protein that can bind DNA and regulate the transcription rate of a multitude of genes. It can thus serve as a master control switch for the coordinated activity of a plethora of genes. The target genes of DAF16s are ultimately involved in the control of metabolism and cell and organismal growth, as well as the regulation of stress response genes and even longevity. While this image shows the IAS signaling pathway as it exists in a typical C. elegans cell, this network also exists in other animals. DAF2-like molecules are generally called insulin/insulin-like growth factor receptors. H1-like molecules belong to a class of proteins called PI3 kinases or PI3Ks, and DAF16-like molecules belong to the FOXO class of transcription factors. We will encounter these molecules throughout this course. The IAF is only one of many pathways identified that are able to modulate lifespan. But regardless of the pathways that longevity genes are part of, one observation that researchers made early on is that many of these long-lived animals show exceptional resistance to a whole variety of stresses, such as heat stress, UV radiation, or oxidative stress. These observations hold true for a variety of C. elegans longevity mutants, as shown in this picture, but also for longevity mutants in fruit flies or mice. This seems to indicate that many pathways that are able to increase longevity may do so by overactivating genetic responses that increase the stress response, leading to increased stress resistance and improvement of homeostasis. So what then are the genes that are regulated by DAF16, FOXO, that enable an organism to become stress resistant? An important clue came from stress-resistant C. elegans mutants that are long-lived and was subsequently corroborated by work in Drosophila mice. Genes that were upregulated as a consequence of DAF16 FOXO activity include GST or glutathione S transferase, superoxide dismutase or SOD, catalase, and small heat shock proteins. So prevalent is increased stress resistance in long-lived organisms that researchers are even using this phenotype as a proxy phenotype to screen for novel longevity genes. The idea is to expose animals to high doses of a specific stressor, sometimes even over multiple generations, and to only select the surviving animals. Since those survivors are stress resistant, the reasoning goes that many also may be long-lived. In this particular experiment shown, resistant survivor cells were then isolated, differentiated into germ cells, and then used to raise new animals, which were then also found to be stress resistant. It bears repeating, though, that as aging is a multifactorial phenotype, not all longevity mutants are stress resistant, and not all stress resistant mutants are long-lived. The other molecules that I mentioned, GST, SOD, and catalase, are part of the cellular defense system that aims to prevent damage to the cellular macromolecules, such as proteins, lipids, or nucleic acids. These systems that GST, SOD, and catalase are involved in protect macromolecules from damage caused by reactive molecules that are generated either as part of normal metabolic activity or in response to external stressors. The GSH system, for example, is the most abundant cellular detoxification system and plays an outsized role in this regard, as it is central to all detoxification reactions. It is based on the small molecule glutathione, or GSH. Glutathione is a small molecule that can be used by the enzyme GST, glutathione S transferase, to neutralize toxic, or chemically speaking, highly reactive compounds that are present in a cell. If they come from external sources, they may be referred to as xenobiotics. Sometimes you also talk about them as toxins. This neutralizes the reactivity of these compounds. They are no longer to chemically react with cellular components and now can safely be expelled from the cell. The other important cellular detoxification system is the SOD catalase system. SOD is an enzyme that can convert highly reactive superoxide radicals, or O2- radicals, into hydrogen peroxide. Hydrogen peroxide still is very reactive and can damage cellular macromolecules, but this in turn is converted by catalase into harmless water. Thus, highly toxic and reactive radicals are converted in this two-step process into a harmless end product. Interestingly, both of these detoxification systems are connected via the enzyme GSH peroxidase, or GP, which is another enzyme that can use GSH, but in this case, it converts peroxide into harmless water. GSH peroxidase thus complements the action of catalase. In some species, GP-like enzymes are replaced by the enzymes peroxyredoxin and thioredoxin, but the same principles still apply. Interestingly, many stressors, including UV light, X-rays, certain drugs, are potent generators of radicals, including the superoxide radical. Since they are creating these superoxide radicals, those agents are said to be creating oxidative stress. Even everyday life events, such as having to prep for a midterm, are able to create oxidative stress. So in molecular terms, this is what stress really is: the creation of highly reactive molecules that can and will react with and damage cellular components. Even normal metabolic processes create radicals. In fact, it is an unavoidable consequence of cellular metabolism. So how does this work? The majority of cellular energy in the form of the molecule ATP is generated in the mitochondria of the cell in a process called oxidative phosphorylation. This process occurs in a chain of multi-protein complexes, collectively referred to as the electron transport chain. As part of this process, electrons are passed through the protein complex of the electron transport chain until they are transferred to their final electron acceptor, which is molecular oxygen. This is the reason why we breathe, to get oxygen, O2, from the atmosphere into our lungs and from there to the tissues where that oxidative phosphorylation takes place. Unfortunately, that process is not completely efficient and is sometimes leaky. So, as an unavoidable side consequence of the electron transport chain, it creates O2 minus radicals, peroxide radicals. If you ever lit a candle or reversely snuffed it out by depriving it of oxygen, you also know that molecular oxygen is highly combustible, or in other words, very chemically reactive. The burning flame is merely a chemical reaction between air, oxygen, and the carbon of the wick. And in slightly different form, it also occurs between oxygen and the cellular macromolecules. Thus, oxidative stress is a completely normal side effect of life and normal cellular energy generation. But these radicals need to be detoxified, and this is again where SOD comes in. Apart from that superoxide radical, cellular metabolism may also create other radical species, such as hydrogen peroxide, H2O2, or the hydroxyl radical, OH-. Collectively, these are known as reactive oxygen species, or ROS. By their very nature as radicals, these molecules contain highly reactive, unpaired electrons, and those unpaired electrons will readily react with electron-dense structures in biomolecules. Electron-dense structures occur in pretty much every biomolecule. For example, unsaturated fatty acids contain double bonds, which are primary targets of oxidation. These oxidation reactions destroy the fatty acid. This is essentially the process of butter turning rancid, destruction of the fatty acids in butterfat into smaller molecules. Since fatty acids are components of the lipids that form cellular membranes, radicals have the ability to destroy cell membranes. Double bonds also exist in the DNA bases and in proteins, highlighting the highly damaging potential of these ROS. Just as they would be damaging lipids, they can similarly damage the genetic information, leading to mutations, or can damage cellular enzymes, leading to functional problems. The realization that ROS are produced as part of normal, unavoidable processes, as part of the energy generation processes in the cell, and that long-lived animals often are resistant to oxidative damage, has led to the formulation of the free radical theory of aging. This free radical theory of aging essentially states that ROS that are produced as a natural byproduct of normal metabolism damage cellular macromolecules. This damage is largely kept in check by the detox systems we talked about. But over time, damage outpaces repair capabilities, leading to deterioration of cellular function, including repair functions, which further accelerates that damage. And the end result is death. This was the first theory to propose a molecular explanation for aging, and we will talk more about it in the next module. Check your understanding. Define homeostasis. Define stress. Explain how researchers were able to relate stress resistance to longevity. Define ROS and oxidative stress. Explain sources of oxidative stress. Explain the free radical theory of aging.